Fermentation in the yeast occurs when yeast eats sugar and produces CO2 and alcohol.
Yeast is a fungus, and just like you and me, it has to eat. After it eats, it produces carbon dioxide gas, just like you and me. The easiest place to see that carbon dioxide gas is in a piece of bread. All these little bubbles that you see in bread are carbon dioxide gas bubbles that were produced by yeast. The yeast in this envelope are in suspended animation. When you mix the powder with water, trillions of tiny yeast cells come back to life instantly. Incredibly tiny. Say you took a piece of hair and you cut it and you looked right down on the end of that piece of hair. This yellow circle is the end of a piece of hair. These little blue circles are yeast cells. You could fit 10 to 20 little tiny yeast
Chemically, agar is a polymer made up of subunits of the sugar galactose and is a component of the cell walls of several species of red algae, and they are usually harvested in Eastern Asia and California. Agar was mostly used as a home for various microorganisms, mostly bacteria. Preparing the agar plates, you must make sure to keep the petri dish cover on. If you don't, airborne contaminants might get inside. Agar is a gel at room temperature at about 65 degrees. It melts at about 85 degrees. Agar gets hard at about 32 to 40 degrees, and that is why we set it in the fridge overnight. Depending on the agar, they will have different degrees of stiffnesses. Here we are making the glass hockey stick to spread the cells along the agar plate to grow the bacteria. The glass hockey stick is made by burning it and melting it into the shape of a hockey stick. We put the plates in the incubator at 37 degrees Celsius. We placed this agar plate underneath the sink to see what would happen. You might wonder why agar is like a gelatin, kind of like Jello. Well, it is used for culturing bacteria. The difference between gelatin and agar is that agar will not be eaten by bacteria like Jello would. It is firmer and stronger than gelatin is. The agar plate that was placed underneath the sink just shrunk and did not grow bacteria. Agar is also known as serving as a thickening ingredient for soups, sauces, ice cream, cosmetics, carbonating beverages, and for making fabrics bigger. If you do not get rid of all the sanitizing solution, then it will kill some of the yeast and make carbonation almost impossible. Carbonated beverages can taste bad if you do not clean your utensils or if you do not refrigerate soon enough. When making root beer with yeast, the yeast eats the sugar in the root beer, creating carbon dioxide. The carbon dioxide then gets trapped inside the root beer, giving the carbonation it needs. If you do not use yeast, the root beer would be flat. The carbon dioxide produced by the yeast will try to escape, but as long as the cap is on tight, the root beer will become carbonated. The carbon dioxide gas causes bubbles in the beverage that make fermentating solution go into the air. This will leave a mixture of ethanol and water. It is extremely important that no air is present during the process of yeast, or ethanol acid, which is found in vinegar, will be found. Putting the root beer in the fridge is the most important step. If you do not refrigerate the root beer soon enough, the yeast will eat up too much sugar and become alcoholic. The reason the root beer has foam is because of the chemical sodas have that produce foam. Without the chemical, you get smooth, creamy flavors.
Yeah, you drink it first.
What does it taste like?
Not root beer. It's like a mouthwash. It tastes like mouthwash with soap. That's what it tastes like that type of soap. Yeah. That's good. Yeah, that drink was bad. It tastes like mouthwash